Good evening everyone. I hope you can hear me. Let me just share the screen. Can you all see the presentation in slideshow mode? Can anybody just confirm? Yeah, thank you. So welcome back to week 5 of NPTEL online course Drug Delivery Principles and Engineering, and we are discussing some few key points as well as the previous assignments. So I'll just move ahead with what all was taught in week 5. You guys must have gone through all the lectures. I'll just give a quick recap. So as you know, we are learning about various drug delivery systems. Like we have in the past, we have seen what are drug polymer conjugates. We have seen hydrogel systems. We have seen reservoir systems, erodible, non-erodible matrix systems. These were all macro systems. And in the last week, we saw what are micro and nanoparticles. These are nothing but the bigger implants broken down into this micro and nanometer scale particles for easier administration. So all of this we have seen in the drug delivery. In this week, we will be introduced to something known as tissue engineering. But before moving on towards the actual what tissue engineering is, there's another important concept that's been discussed all throughout the lectures in this week, and that is protein adsorption. So what does protein adsorption mean? It is nothing but adsorption of proteins on any surface. Now why this happens is because, particularly why we are learning about this is when you, let's say, you're doing any drug delivery or you're doing any tissue engineering, you most of the times have to inject something in the patient's body. Let's say it's an implant, or it could be any drug delivery system that we have discussed. So when you do that, it has a surface over it. Let's say an implant has a surface over it. So once you inject it into the patient's body, what's going to happen is there are a lot of proteins in a human body, and they since they are weak surfactants, they quickly adsorb over the implant surface, and this then drives how the implant is going to stay in the body, if there is going to be any immunological reaction, how the implant is going to behave. So that is why protein adsorption becomes a very important topic to know. So yeah, as I said, why proteins adsorb on the surface is because they are weak surfactants. And so basically what happens is when you place a material inside a human body, first of all, what is going to happen is there is a lot of water content in the body. So first the material will interact with water, and then it will be followed by proteins, and then lastly the cells that are present in the patient's body will then start interacting with the material. Now what does this tell us? That what cells are going to interact with the material is going to be governed by what protein layer it has over it. So this essentially means that the adsorbed protein layer mediates a biological response. So you can make use of this information to then, let's say, depending upon your application, if you want some particular cells to interact with your implant, then you can code these materials with that particular proteins which are very compatible with the particular cell. So now why this happens is, why will cells interact with this? Because cells have something known as integrins. These are the transmembrane receptors present on every cell. So these integrins then in the natural process, they are particularly there because they interact with the ECM proteins. ECM is nothing but the extracellular matrix that is present outside the cells. So it has a lot of biological functions in signaling as well as in movement. So it's essential for the cells to interact with this ECM via these proteins. Now these proteins, as I said, when you place a biomaterial inside the patient's body, the proteins are going to cover it, and then your cells are then going to interact with the this particular material. So as I said, depending upon the application, let's say you have implanted something in the patient's body and you don't want more cells to interact with it. You don't want any immunological reaction to happen. So in that case, you can coat your material with a protein which the cells are not very they don't adhere to this protein very much. They are not attracted towards this protein. For example, albumin. On the other hand, if let's say you have implanted something where you want tissue integration, you want the cells of your body, let's say it's a bone implant, so you want the bone cells to interact with this particular implant, and you want that implant to be integrated into the patient's body. In that case, you can coat it with proteins which will then give appropriate signals to the cells, and then more and more cells will come and interact with the material. So that's why this concept becomes important whenever you're going ahead with any tissue engineering or any drug delivery system. So yes, and in the later lectures of this week, we were introduced with tissue engineering, which is nothing but using cells, materials, and engineering principles to improve or replace biological tissue. Now, why do we need tissue engineering? This is nothing but making an artificial organ or making part of an artificial organ and replacing the damaged organ in a patient's body, so that the patient feels better and the function of the damaged organ can be replaced. Now, why do we need this? Because right now we have the way of transplanting is still there, where you have human donors, and from these donors and the appropriate organ is extracted, and then it is implanted into the patient's body. But then this has a lot of risks, like it could spread disease, and there's always this risk of immune rejection, because even though we are of same species, no two humans are similar, and taking an organ from one person's body and implanting it to another will definitely have immune rejection. And there is also this problem of lack of donors, because not enough donors are always present for to treat the patients. So this is why we need artificial tissue engineering, and there are various ways in tissue engineering works. First of all, it could be used to generate a whole tissue in vitro. That is, you completely make an artificial organ outside the patient's body and then replace the damaged tissue in the patient's body. Or you can make part of the tissue in vitro and then replace the damaged portion. This happens in case of, let's say, there's a liver transplant. So liver has a lot of regeneration capacity. So let's say part of your liver is damaged, so you don't have to replace the entire liver. You can just replace the damaged part and then take advantage of the liver's regenerative capacity to then come back to its normal function. Or it could be putting scaffolds to regenerate part of the tissue in vivo. Let's say you have a scaffold and it doesn't have any cells. Scaffold is nothing but just a pattern of how that organ is, and then this is just the ECM of that organ, and then you replace it in the patient's body, and then over time the body cells will start integrating into the scaffold. They will start building over it, and then you can regenerate the tissue in that way. This is completely done in vivo instead of making the organ in vitro. Or it could be adding support material to support the damaged tissue. For example, let's say there are bone implants. So in this way, tissue engineering can work. So these were some of the key points that were discussed in week 5 lecture. So with this knowledge, we'll then go ahead with the previous assignments and try to solve some of the questions. Does anybody have any doubt here before I move on towards the questions? No? Okay, then I'll go ahead with the assignment question. So I hope you guys can see the question displayed on the screen. So first question is, why do proteins adsorb on material surfaces? And we have to pick one option. There are several options here. First is because they are weak surfactants, or because they tend to increase their activity after interaction with the biomaterial surface. It could be to prevent biofouling on the surface, because they are strong surfactants. The proteins will never adsorb on material surfaces, or it is to decorate the surface. So we have to list a reason of why, first of all, proteins adsorb on material surfaces. If anybody wants to volunteer for the answer, you guys can go ahead, unmute yourself and let me know the answer. Somebody is trying to answer in the chat, so he's saying option 1. Does anybody have any other answer? No more response? Okay. Yeah, whoever answered, you were correct. The right answer is first option. As we were just discussing in the previous slide. So why do proteins absorb? Is because they are weak surfactants. Now what does this mean? Weak surfactants meaning a protein will have both hydrophilic as well as a hydrophobic part in their structure. So whenever you implant any material gets exposed in the body, these proteins can then easily adapt its shape to interact with the material. Let's say you have a material that is very hydrophilic, then the protein's hydrophilic parts will quickly interact with this. So the protein's hydrophilic parts are mostly on the surface of the protein, so they will go and adsorb to the particular material. If your material is now hydrophobic, the proteins will then unfold its hydrophobic core parts which were hidden inside, will then like to interact with the material because there's a lot of water layer around it, so they will try avoiding the water, and then the hydrophobic parts can then interact with the material surface. So in this way proteins adsorb to the material. So this is essentially because they're weak surfactants, they can easily change their shape, change their structure to adhere to the surface. Is this clear?
Yes, ma'am.
Okay, thank you. Then I'll move towards the next question. Yeah, so the next question is, which of the following two statements are false about protein adsorption in biomaterial? So we have few options here and we have to select two of these which are false about protein adsorption phenomena. So read through the options carefully, take your time, and if anybody wants to answer, you can start answering. I'll wait for a few minutes.
Ma'am, option 3 is wrong, I think, because
Okay.
they can come off when when there are more proteins present in that surface. Other proteins can come and adsorb, whereas some of the protein they might come off.
Okay, sure. Okay, so you're saying option 3 is one answer. Do you have any other option? Because we have to choose two. Okay, Suhail is saying 1 and 3. So 1 is limited absorption sites and consequent competition means that not all proteins in the plasma phase can be represented on the surface, and this is third. Okay, so we have two answers here, 1 and 3. Anybody still wants to volunteer? So we have to choose two false statements about protein absorption. No more response? Okay.
Ma'am, I think 6 because proteins don't always lose their structure and function after adsorption.
Oh, okay, this one, the last second. Proteins always lose their structure and function after absorption. So you're saying 3 and 6, right? Okay.
Yes, ma'am.
Okay, so we have two answers, 1 and 3, 3 and 6. Okay, then we'll look at the answers. So the correct answer is, you're correct, third option and the sixth option. So we'll go through the options one by one. So first is limited absorption sites and consequent competition means that not all proteins in the plasma phase can be represented on the surface. So this is actually correct, this is not false. So whenever, let's say, you have a biomaterial that's implanted, and it is going to have a very finite surface, and there are lots of proteins present in the human body, in the plasma. So not all proteins could be represented on the surface because there is very limited space and there is a lot of competition going on. Some proteins might be might have greater affinity for this biomaterial compared to others. So they will quickly absorb on the surface and not leave space for the other proteins. So that's why the first option is correct. Driving forces for absorption are the intrinsic surface activity and bulk phase concentration of the proteins. This is also correct because the surface of the biomaterial is very active and it can interact with the proteins, and since these proteins are present in bulk, these are the driving forces for the proteins to absorb on the surface. Third is, once a protein gets absorbed, it doesn't come off, is actually false. So this attraction of protein and the surface is a physical attraction. There is no chemical bond involved. There is no covalent bond involved. This is just because of its hydrophilic or a hydrophobic nature. So whenever a protein gets absorbed, it can easily come off. It's not like a very permanent interaction there. And then surfaces vary in selectivity of absorption is also correct. Different surfaces will have different selectivity of absorption. Not all surfaces will absorb a particular kind of protein. It will depend upon what kind of surface is it. Absorption is highly dynamic. This is also correct. Once a layer of proteins has been absorbed to a surface, it doesn't necessarily stay that way. It will keep on changing based on what proteins are now interacting with the surface. And the sixth option is, proteins always lose their structure and function after adsorption, which is also false, because not necessarily, let's say there's a hydrophilic protein, and as you'll know, for proteins, the hydrophilic part of the proteins are on the surface and the hydrophobic core is inside. Now, if you have a biomaterial that is hydrophilic, the proteins will just go and adhere. It will just like sit on the biomaterial. There's no reason for the protein to lose its structure and function after adsorption. On the other hand, if there's a very, very hydrophobic surface, then the proteins will then lose their structure. So all the hydrophobic part which is buried inside will come out. In that case, the protein has to lose its structure and consequently the function. So it's not always the case that every protein is going to lose the structure and function after adsorption. It depends case on case. And then biological activity of the adsorbed protein varies on different surfaces is also correct, depending upon, as I said, what interaction it is, what kind of protein it is, what kind of surface is it. The biological activity will then be different. It's not like all proteins will be bioactive or they may not be. So that's why the false statements about protein adsorption are these two. Once a protein gets adsorbed, it doesn't come off, is false. Proteins always lose their structure and function after adsorption is also false. Is it correct? I have put the explanation from the lecture itself in this. You guys can read it later on. So I'll be sharing this PPT as well. Is this question clear? Does anybody have any doubt with the options? Or somebody else is also trying to answer. Okay, thank you. I can't keep on always looking at the chat box, so I have to flip between the screen. So just bear with me if I'm not acknowledging your answer. Can I move towards the next question?
Okay. Yes, ma'am.
Yeah, thanks. The next question is, consider that cells are resuspended in a rich protein solution, let's say FBS. The suspension is added on top of a biomaterial. Now we have to rank the interaction of different components, which could be cells, water and proteins, with chronology of their interaction with surface of biomaterial. I think we discussed this in the first slide itself, so should be easy to answer if anybody wants to volunteer. So it's nothing but just where your biomaterial is placed in FBS, and we have to rank what is going to interact with the surface first.
Ma'am, it's slide 1B.
I think two people answered the same, third option, right?
Yes, ma'am.
Okay.
Yes,
Water,
ma'am.
protein, cells. Thanks for answering. Anybody else has a different sequence? third. Yeah, all of you are correct. Pretty straightforward, as we just discussed that the way things will interact with biomaterials, because it depends on the abundance of these things. Actually, if you place a biomaterial in a solution, most of the solution in the body, or let's say in this case FBS, it's going to be water. So it's easier for water to first interact with the surface. Then proteins are more in number compared to the cells, so proteins will then interact, and then depending upon what proteins are interacting, your cells will interact with the surface. So yeah, this is pretty straightforward. I'll move towards the next question. This question is, what are the common techniques to quantify protein absorption on material surfaces? And we have to choose two correct options. So this has more to deal with the techniques. So,
it's rest and surface plasmon resonance.
okay, I couldn't hear your first option. Surface plasmon resonance and
Western
Western blotting. Okay. Is anybody else also trying to answer? I can hear two voices. No? Okay. Thanks for answering. Anybody else? No? Okay. Yeah, you're correct. The correct options are Western blotting and surface plasmon resonance. So both these techniques are used to first of all know if there is any protein absorption on the surface, and then next to quantify it. So it could be done by this method. So first you incubate the material with your, let's say, FBS or any serum. Human serum also could work if you want to see what proteins in the patient's body are going to interact with your surface. So you incubate that. Incubate meaning you just put that material in the presence of this serum and allow it to interact, allow all the proteins to absorb to the surface, and then you centrifuge. You separate out the proteins that have absorbed on the surface, and then you run a SDS page gel. And once you do that, you can then follow it up with the Western blotting technique, and then know what all proteins are there. So you have some standards of proteins of which you know the size, and then you can compare what all proteins that you have gotten. That is one method, that is Western blot. And then next you have surface plasmon resonance. So this is a technique wherein you have, let's say you have a surface, and this is your biomaterial surface, and you coat it with some metal, and then you again incubate it with, you allow the serum proteins to interact with this with the surface, and depending upon what proteins are interacting with the surface, the dielectric of this particular surface changes. So to measure how exactly is the dielectric changing, you can then take a light source and you can shine it on this surface. So in case there are no proteins, it will have a particular angle of reflection. Once you have proteins interacting with the surface, this angle of reflection then changes. And then you can run some standard solutions. You already know what proteins are there and what is the quantity of these proteins. And in that way, you can have a standard curve, and then you can then compare with what your sample proteins were and how much was the concentration of these proteins. So these are few techniques to know if protein absorption has happened to the surface, and then quantify, and in some cases to know what protein these are. So yeah, these are two techniques. Clear? Yeah, somebody else also answered the same thing. Thanks for answering, people. Okay, I'll move on towards the next question. The next is, what is Roman effect? And we have to pick one answer. Few options here. Go through the options and then we'll discuss.
Second one.
Second is replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. Okay, thank you for answering. Anybody else?
Option 2.
Okay, thank you. Anyone has a different answer? Okay, so I'll discuss answer. Yeah, both of you are correct. The correct answer is replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. This is known as the Roman effect. So what this essentially means is, as I said, the absorption of proteins onto a biomaterial surface is very competitive. So first of all, what happens is when you biomaterial gets exposed to these number of proteins, initially the kinetically favored, meaning if there's a particular protein that is present in a very high concentration, naturally, since there are more molecules of these proteins, they can quickly come to the surface and take much of the space of this surface. So these are known as kinetically favored, initially absorbed proteins which are kinetically favored. Then what happens over time is this interaction of these initially adsorbed proteins and the surface is not very strong. So later on, let's say there is another protein which is present in less concentration compared to the first one, but it has a very strong attraction towards the biomaterial surface. So these proteins can then come in, they can displace the initial layer of proteins and then adsorb to the surface, which will then have a very strong interaction and have a so they can stay on the biomaterial surface for a long period of time. So this is known as the Roman effect, wherein, as the option says, replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. So yeah, that's what Roman effect is. This is clear? You people can let me know if I'm going too fast and then I can slow down. Thanks for answering in the chats as well. Okay, I'll move on towards the next question. The next is, when nanoparticles are injected in vivo, the proteins form a layer on the surface of the particles. What is this particular layer called?
Protein corona.
Okay, option 3. Thank you. Anybody else?
Option 3.
Okay, thanks. Anyone has a different answer?
Three.
Sorry?
Yes, ma'am.
Okay, yeah, I think you also answered the same option. Others are agreeing with the answer, I assume. So yeah, all 3 of you are correct. The correct answer is protein corona. So as adsorption can happen on material surfaces in plants, it can also happen on nanoparticles, because proteins are very, very smaller in size compared to these nanoparticles. So what happens is, when you inject nanoparticles in the patient's body, there's this corona layer that forms. Initially some proteins will come and absorbed over it, and this is known as hard corona because it in some sense is a very stable layer, and then you have this soft corona surrounding this hard corona layer. So the soft layer can be very dynamic in nature. It can so the proteins present in this layer, they could interact with the hard corona layer, and it's not very stable. It's not a very strong interaction. These can come and go as their first of all their interaction governs and their concentration as well. So these two layers then form a hard corona and a soft corona. So essentially all of this is known as the protein corona over the nanoparticle. Clear? Okay. The next question is, if cells are transplanted from a human donor to a human acceptor, what kind of transplant is it?
Allogenic.
Okay. Thanks for answering. Anybody else? Okay. Others, any different answer than option 1? Okay, most of you are answering is option 1. I'm assuming it's for this question and I didn't miss the previous question. Yeah, I'll discuss. All of you are correct. The correct answer is allogenic. So there are different terms for this transfer that takes place in between different species. So allogenic is taken from different individuals of the same species, which means any transplant happening from human donor to a human acceptor. Autogenic is taken from the same individual. This happens in case of stem cell therapy, where the doctor will isolate stem cells from the same patient, engineer it for some particular application, and then implant it back into the same individual. So this is known as autogenic. And xenogenic is taken from individuals of different species. For example, let's say there's a pig implant or a liver taken from a pig which is then implanted into a human. That kind of transplant is known as xenogenic. Antigenic has nothing to do with transplantation here. So yeah, the correct answer is allogenic. Moving on towards the next question. Next is, for transplantation in humans, allografting is one of the most common approach. What are the major drawbacks of this approach? We have to select 2 correct options.
Option 1 and 2.
Yeah, one by one. One person said 1 and 2. Anybody else also trying to answer?
Option 1 and 2.
Okay, thanks for answering. Any other response? No? Okay. Yeah. Both of you are correct. It's because of the major drawbacks of transplantation in humans is lack of donors and spread of disease, like we also discussed. There are more amount of people waiting for getting an organ transplant compared to the donors. So first drawback is lack of donors, and then it could spread the disease if the characterization of a donated transplant is not done, or if there is very less time, the patient's life is on the line. In that case, you have to completely speed up the transplantation process. In that case, if the characterization of the donated organ is not done in a very good manner, then we could skip over some of the diseases that organ could carry, and then it could spread the disease. And lastly, the drawback is immune rejection, as most of the half-life of half-life of most of the organs that are transplanted is around 10 to 15 years. By this time, then the patient's body starts rejecting the organ because it's in some sense still foreign. So yeah, the major drawbacks are lack of donors and spread of disease. Rest of the options don't make sense because lack of oxygen in operation room is not the correct answer. Organs can stay functional for months outside the individual is also not correct. Doctors are reserved when it comes to harvesting organs is also false, and current technology cannot support organ replacement surgery is also false. So the only two drawbacks for this particular transplantation is the first two. Is this clear? Okay, I'll move on towards the next question then. Next is, which surface is more favorable for protein absorption and why? We have to pick two.
First option.
2 and 4.
Okay, one person said first option, hydrophilic surface, because it is more charged, and another person is saying 2 and 4. Okay, anybody else? Thank you for responding. Any other response? No? Okay. Yeah, one of you was correct. The correct answer is hydrophobic surface because it is energetically more favorable to displace water and replace with protein, and then a rough surface because of available surface area. So this has to deal with how proteins interact with the surface of biomaterial. So essentially what happens is there are different surfaces which would have different favorable conditions for the protein absorption. So first is hydrophilic surface. So in that case, if your material is hydrophilic, there is a lot of water molecules also present in the patient's body, so they can easily interact. So that doesn't favor protein absorption to that extent. Now in another case, if your surface is very hydrophobic, your surface will try to exclude water. It does not like interacting with the water. So what will it do? Since it's hydrophobic, it would like to interact with hydrophobic proteins. So in that case, that's why protein absorption is happening more on a hydrophobic surface, because it is energetically more favorable to displace water and replace with the protein. Then if you have a smooth surface, so the first two options have to deal with whether your surface is hydrophilic or hydrophobic. Next two options are dealing with whether your surface is smooth or rough. Now if you have a rough surface, you will have a lot of surface area. So rough surface meaning instead of having a smooth line, if you have, let's say, some pattern like this, zigzag pattern, so in that case that particular surface will have more surface area for more and more proteins to come in compared to a smooth surface. So that's why if you have a rough surface, it is more favorable for protein absorption. So these are the two reasons. So this becomes important whether when you're trying to build up an implant or you're trying to tissue engineer, right? You can then tune around what surface you want, whether you want more protein absorption. In that case, you can go for a hydrophobic surface, you can go for a rough surface. In case you don't want much protein absorption, you can then go for a hydrophilic and a smooth surface. So is this clear? I think this was the last question of the assignment that I have for you today. If there are any doubts, I can discuss. If anybody has missed any previous questions or wants me to repeat the explanation of few of the questions, we can go over that. But yeah, so that's all I have for you. I guess. Yeah. Anybody has any doubt?
I have a query.
Yes.
I was not able to attend the Saturday's live session because I was not able to join for the
Okay. Okay. Yeah, because my session. Oh, Saturday session. Okay.
this one session. Yes,
Okay.
I was asking to join many times, but I was not let, and I was not sure what is the reason.
There
So
could be some
it's like
technical.
I have been trying for long.
Yeah. This is the Yashwant session, right? The Saturday session. This is taken by another
I
TA.
guess
Yeah. Yeah.
I guess
Okay. So, is there anything you want to discuss from that
yeah.
session?
Nothing is specific because I couldn't attend that session.
Okay.
Anyway, I can check with the recorded
Yeah.
session.
Yeah. It
Not an
would be
issue.
uploaded on YouTube by
But
now.
yeah, but if there's any technical glitch, so I just thought of reporting it to you.
Sure, sure. I can talk to Yashwant. There might be some problem in case with the Wi-Fi that we have here. So I'll just check with him and let him know that few people were facing problems while joining. Is that okay?
Sure, thank you. Yeah, thank you.
Welcome. Any other doubt?
Ma'am, I have a doubt actually regarding the week 3 assignment.
Okay.
Actually, in one of the questions,
Yes.
in 9th, though I marked the right answer. So I think like the previous assignment 1, I think it was updated laterly, and then I think everybody got the right answers, also the score.
Right,
So
right.
I think week 3 also needs update, and the matter should be resolved.
Yeah, sure. So yeah, I think I remember a few of you telling me about the first two assignments as well. There was some problem with the multiple choice questions. So what happens is when you answer, it takes some time for the TAs to get the assignment, and then they can go ahead with the checking. So if there's a problem with week 3 assignment, I think you'll have to wait for a week or so till that gets resolved. But don't worry, your grades will be updated immediately after it's checked, so it won't hamper your gradation. I'll let people know that there's some problem with the week 3 as well. Is that okay?
Thank you.
Yeah. Any other problem? If not, then I can close this meeting and we can meet next week with week 6 points as well as any questions that you might have. Okay, you're saying even in week 1 assignment, question 9 was not correct. Okay, I think that has been resolved by now. You can just check and let me know. If not, then I can talk about week 1 assignment also. Apart from the assignment thing, any other doubts?
Ma'am, I have one doubt. Like for there are questions where we have to choose two options.
Okay.
One of the question I didn't I did not notice that we have to opt for two options, but I have selected for one option. They have given me 1.5 marks for that.
Okay.
Whereas in another question, after that I got to know where there will be like we have to choose more than one option.
Right.
The next week there was a question like where we have to choose two options.
Okay. I think
Correct. The other one is wrong. Is there like if two are then only the marks will be awarded, or if one is correct, they will award the marks, or
Okay, so as far as I know, I think if you answer one correct, you will get half of the marks. If you answer both the options correct, you will get full marks. If either of these options is wrong, then I think you lose all the marks there. I think that's how the assignments are graded, but I can check with them whether it's correct that even one option is wrong, then the entire question gets no marks. So yeah, I can check and let you know.
ma'am, in week 2 and week 4, they have not awarded the marks for me in that type of questions.
Okay, I'll check. I think week 3, week 4 they have to anyways go over again because most of the people are facing problems. So I'll check and let you know. Yeah, Bhavadharani is saying that for me the mark was not awarded. Okay, I will ask them to check again. Is it still in the week 1 that you are facing this problem? Okay, sure. I'll check with the TS, the current TS that are checking your assignments. Any other doubts here? Okay, Suhail has a doubt saying, does the charges of the proteins play a role in protein absorption? How does this displacement of proteins occur? There ain't any attraction or repulsion between the proteins. Yeah, so charge of the proteins will also play a role, and there definitely is attraction or repulsion between the proteins. Let's say for example, the first proteins that have adsorbed onto your surface are positively charged, and another protein that is also positively charged is trying to come near the surface, and in this case it will be repelled by the layer that is already formed on the biomaterial. So charges as well as the nature of these proteins do play a role. So these charges have to be at the physiological pH. So your protein could have a positive charge outside the body, but inside the body at pH 7 we need to see what is the charge of these proteins, and then that's how the interactions get governed. So this displacement of proteins is nothing but, let's say you have this is simple adsorption, right? You have a surface, one protein is coming and attaching here, another protein that is more strongly attracted towards the surface will completely displace this because the interactions with the protein and that surface are weak. That's why the protein will get displaced. I hope I am able to answer you. Okay, if you still have doubts in this, I can again read more upon this protein adsorption and come back in the next lecture, and we can still discuss if you are interested in knowing more about this. Thanks. Others still in the meeting, do you have any questions? If not, then I'll stop the meeting here. Thanks for joining today. See you next week. Thank you. The rest of the two people, do you have any doubts, or shall I end the meeting? Okay, I'll stop it here then. Thanks for joining.